Good evening, everybody. Why don't we get started? I'm Dan Parsley. We have, we'll make this a fairly brief call, but I wanted to introduce Zafar Syed, who is the CEO of Saranas, who will be briefing us on the update on Saranas, some very exciting developments, and then specifically talking about the new $2.8 million raise that they've opened, that we have the opportunity to invest in, and then give you the opportunity to ask any questions. We're on as investors in Saranas from our syndicate that we closed just under a year ago in May of 2017. You have the rights to reinvest in Saranas in this current round, and so you will have the opportunity to do so. In fact, you'll have a chance to invest either minimally up to the amount to maintain your ownership percent, but also opportunity to invest above that should you choose to. We are on a fairly tight schedule with Saranas, and we'll ask you to please make your decision by Friday of this week so that we can give our existing investors the first opportunity to take the amount allocated to us, which, Zafar, perhaps you can confirm exactly what that amount is when you make your presentation. So for what is not taken by the existing investors, then we will open that syndicate up to other investors. And so one thing I would just confirm as a reminder, the syndicate that we closed last year that you invested in had a post valuation of roughly $12 million. And we indicated at that time that there was a good possibility of a 12 to 24 month exit of Saranas at a exit range that we had estimated to be about $50 to $100 million. And I think you'll find with this round that Zafar is about to introduce, this round has a post valuation now of roughly $32 million, so it's a nice step up from the previous round. But we are estimating that the exit valuation of the company is a $150 to $200 million range, which is a nice appreciation in the estimated exit from just a year ago. Certainly makes your previous investment that much more valuable, but still leaves a very attractive investment in this round, given that the post valuation is 32. So I will stop there and turn it over to Zafar, who will give you an update on the company and more details on this race. Zafar.
Thanks, Dan. So thanks for the intro. I'm going to go ahead and I'll try to track to the attachment that John had to the call so you all can kind of follow, and it gives a nice summary of an update of our achievements and really what we're trying to plan for this particular raise. And then, I've been told, we want to keep this open for questions as well. So, I'll keep it relatively informal. So if there's questions, even along these slides, I know there isn't a lot of people on the call. It might be easier to facilitate that. So feel free to ask me questions during the presentation. So I'll go ahead and track to the presentation that is attached. We are planning a 2.8 million Series C raise, and really the objective at this point is to try to collect clinical cases that helps validate the clinical thesis that we have with this particular device, and I'll share some exciting data on our animal study where we feel that the performance is going to be hopefully replicated in the clinical pilot. We're also part of this raise preparing ourselves if there's not any deal making that's going to be happening while the device is being reviewed by the FDA and while we're conducting these clinical pilots, to really be able to exercise the business model. And I think we'll make an argument, at least with the installed bases in our clinical pilot sites, on how we're going to be able to strategically build that out, and I'll be able to speak to that. And really, we want to use the Series C funds to really maximize company valuation for a successful exit, and that is particularly in demonstrating the clinical use case with the clinical pilot. The scope, as has been identified before, is since we closed in the last Series B, trying to exit within 12 to 24 months. We're still within that time frame, and I think we've been able to make some significant movement to drive awareness amongst the strategics. And really, we're trying to minimize any kind of additional expenses within the team. We have a pretty lean team right now, and as we kind of move toward this next phase with the round, we're going to continue to do that. But we are going to leverage some consultant help on driving some of the clinical activities. So as I kind of go along and look at the milestones that we've been able to achieve over the course of literally less than 12 months, since taking on our Series B, we've had some significant traction, particularly around completing our early bird product development, which includes passing a final animal validation study protocol, and I'll go into the details of that, where we actually exceeded our expectations on how the device was performing. There was also a significant move in how we are positioning the device to be used as a bleed monitor, with the use case of placing this now as an auxiliary monitor in the femoral vein, which wasn't identified when we first closed, and I think that was from feedback that we've had with our chief medical officer, as well as some testing that we performed demonstrating that it could be used as a bleed monitor to support any potential arterial bleeds that are occurring during the procedure, and even keeping it in the vein for post-procedural monitoring in the femoral vein. We've conducted a successful user validation study with both our interventionists that have been using the device over the course of the few months in our animal study, as well as an independent usability study late in the fourth quarter. We've attended multiple meetings internationally and been recognized as a top cardiovascular innovation at 3 of these international cardiology meetings over the course of the past 4 months. And we've actually enhanced our engagement with our scientific advisory board on top of that. So that's really kind of serving as a good springboard as we move into our clinical pilots. And quite honestly, we've confirmed interest from 10 U.S. centers to participate once we get the protocol finalized and ready to go. And we're on track to submit our FDA de novo submission in the next few weeks. So we're probably about 75 to 80% complete on our content of our packet. We're highly confident with the animal data that I'll be able to share with you that we will get FDA clearance. Obviously, it's going to take time for the review cycle to begin with the FDA, but we anticipate with the data that we have and the final device testing to hopefully achieve FDA clearance by the end of the summer. But we're about just almost weeks away from submitting that to the FDA. And we've had some interesting publications that have come along the way that have supported the data to correlate bleed risks with some of the interventions we're targeting. So we're going to capitalize on that. So on to the animal validation study. So what I do want to highlight specifically is a big milestone is since we stabilized and de-risked the technology of our device in the middle to the late last year, we were able to execute. I'm sorry? Okay.
I'm here. Go ahead.
Oh, okay, no problem. Execute an animal validation study that was statistically sampled, and this was a design of the protocol that was reviewed by the FDA prior to its execution, and it required 40 data sets to achieve 90% sensitivity in order to pass the study, and we actually exceeded that with 100% sensitivity and 100% specificity in the animal study. Here's a sample one of the legs that we evaluated in the pig model. As you can see, the device has very stable impedance baseline in the beginning before we do a controlled infusion of a bleed in our model. You will see that the impedance does fluctuate, which the device appropriately doesn't trigger. These were in the protocol incorporated in order to simulate actual PCI simulations through the sheath and even in the arterial sheath. And it will sense these changes in impedance that might impact the signal, but it doesn't detect those as a true bleed, and so which we were happy to report. And then as the infusion of the blood was fed into this particular model, we were able to demonstrate appropriate detection at various levels of the bleed indicators on our user interface display. What was also compelling is really the story that we're trying to tell, and the data that we were able to show, not just in terms of level 1 bleed indications being highly sensitive and specific to the bleed, because there was a baseline of no bleeds that were designed into the study. We were also able to demonstrate that the subsequent indicators that identify a level 2 or a level 3 bleed had significant separation between each level, which was analyzed and showed to be statistically significant, which actually demonstrates that the algorithm that we have actively detects bleed progression. And that's really the story we're trying to tell. After going through the animal study, there's questions on how much volume is clinically significant. We're able to show that there was clinically significant levels of bleeds that happened between each level, and the story is going to support the idea that the device can not only sense a bleed, 100% of the time, or highly sensitive, but can demonstrably show active bleed progression, which would then allow a clinician to make a decision. So that's very good. We're actually in the process of submitting an abstract to a cardiology conference for this summer, which we're also hosting a symposium that'll highlight our animal study data, as well as submitting it into a journal article by the end of this month as well, hoping for publication to try to time it around the time of the cardiology conference, which is actually the TBT conference in the end of June. So that's kind of where we've gone and kind of where we are right now with our pending FDA submission. But really, what are we doing now? So this particular raise, again, is focusing on us being able to execute a clinical pilot and really enhance our commercial readiness. And we've been able to have very strong conversations with strategics, with clinicians on the clinical utility of the device ever since we've kind of moved to it being placed in the femoral vein. And that's where we've gotten a lot of this recognition. We're building on that with the clinical protocol that we're hoping to launch as a non-significant risk at about 5 to 7 centers in the next couple of months, trying to target for first in human cases in May. As I mentioned, we've solicited interest from at least 10 centers now. We'll have to probably pare that down to the ones that would fit the bill. But we're excited to finally get this device into humans and actually see and learn how the device is going to be able to react to some of these bleed complications that may happen with these types of large bore procedures.
Zafar, I have a question for you. When you do the human testing, are you introducing blood into the area to test it, or are you just testing it on a normal procedure to see if it's detecting it in a normal course of action?
In a normal procedure, I don't think it would be very difficult to try to induce a bleed anywhere. So what we're planning on is identifying or risk stratify the patients that would be at risk of bleeding. So that's why we are trying to enroll a high number of patients, looking at 100 patients across these centers. Now, it's safe to say that we're not, clinically significant bleeds are, published at a rate of about 18 to 20% if you specifically look at large bores, which we'll be satisfied with. If we can get 18 to 20 cases that have clinically relevant bleeds that can be detected by the device, we'll be able to illustrate that in the cases. But I also have a sense that our device is pretty sensitive, and so it'll be interesting to see, what that rate may be in these kind of real-world examples. We're not going to induce a bleed, so we're just trying to see how the device operates for the targeted procedures that we feel that this device is going to be integrated into.
Great, thank you.
And we're going to also, I don't know if I have details of the study here, but we're also going to try to verify whether a bleed occurred or not for hopefully almost every patient with a post-procedural CT. And we're working with a core lab to possibly even develop a scoring matrix that allows us to quantify the level of the bleeds. But that's more on the research side of things. But for us, quite honestly, is identifying if the device is able to trigger upon a potential bleed, and then maybe and being able to at least verify that with a CT is what we're hoping for. And then the idea is to try to enroll as many cases as we can before our symposium, which we're sponsoring in June with a couple other strategics on bleed management at TVT, and hopefully we'll have some cases there to highlight how the device not only work, but maybe some of the options that the clinicians use to triage the issue if the patient did indeed have a bleed that was appropriately detected by the device.
Can you give us an idea of the breakdown of how the $2.8 million is being used?
So the primary use of the funds initially is to drive the clinical pilot activities. So that includes hiring a CRO, understanding what are the costs to support the site expenses. So we believe that about almost, and also, and that's going to be anywhere from $600,000 to $700,000. We're also planning to get our device ready for manufacturability. So through the design process, we've already gone through the development phase, what we call our phase 5, which we're actually doing our final design review in the next couple of days. And that will allow us to get FDA submission and moving towards clearance. But the FDA does also want to have our manufacturing processes validated. So part of that money is really going to also drive the validation activities that are going to be needed at the contract manufacturer level. So by the time we get FDA clearance and ready for potential commercial launch, or even if a strategic is interested on taking on the manufacturing, we will have those processes validated. And a chunk of our expenses, a little bit over $650,000, will be supporting that effort. So what we think is we'll be, up until from now till the end of September, we'll have at least $1.5 million of that money is going to support the clinical pilot activities, as well as being commercially ready for building out commercial product. And then we have other uses of the expenses to support our market development efforts. So the symposium that we're planning for in the summer, we are planning a couple other promotional activities prior to FDA in terms of highlighting some of our milestones as well. And then we plan on supporting our commercial launch if we have to with some of the other market development activities, such as advertorials and white papers. So there's some marketing effort and money that goes through the course of the next few months leading into a potential fourth quarter launch. And then finally, the last portion of the money is to facilitate builds if we have to go ahead and launch at these limited centers of excellence. We're trying to cap the number of centers. I would say it'd be anywhere between 10 to 20, and we would start with the installed base of the clinical pilots. Oh, by the way, there is also one pricing study that we will also be conducting prior to the completion of our clinical pilot, just so we have some more data to support our pricing strategy with this product. Any other questions?
Any questions for Zafar?
Hey, hello, can you hear me?
Yes.
Hey, this is Joe. Hey, listen, I have a question. What's the false positive rate that the studies have shown so far on this device in the animal studies?
So that was the thing. So the way we designed our animal study is we had a 10-minute baseline. So in order to possibly, falsely detect or miss any detections of bleeds, so we induce a bleed in our model 10 minutes after that baseline. But within that 10 minutes, there's manipulations that are done just as, any kind of, cath procedure. So we'll have, wires and catheters kind of funneling through both the arterial sheath as well as our own femoral vein. That's where it was placed in with the early bird. And we didn't get any false positives out of the 40 samples that we conducted in our study. Now, I will tell you, there were studies that were done, in different iterations, but we basically froze our design for this final animal validation study. And with the design that we had, there were no false positives. There were some bleeds that were detected during that 10-minute baseline period, but after necropsy, we identified that, the interventionist actually poked through the artery or what have you, and there were actual bleeds. It was true bleeds that were actually pooling around that access site. So, I'm not, ashamed to say this, but it's 100% sensitivity is what we've identified, 0 false positives. And we didn't miss any bleeds as well.
And so I guess the this when I was talking to Dan about this investment about a year ago or so, was there some for this new ask of 2.8 million? Was there some change in I guess the attitude of this product out there in the market? Because did you guys have initial expectations to go ahead and go straight to the market after the original studies, but then from the manufacturers came back and said, no, we want you to show this, actually prove there's some efficacy with it?
From the strategics? Yeah, I think there's a couple of things. So there wasn't as active an engagement with the strategics. Again, prior to my time coming here, there were some conversations on what we're trying to do, and a lot of the questions that came back were two things: is are these bleeds frequent enough, and are they impactful? And we ended up having data that supports kind of the impact on mortality and cost. I think after those conversations with the strategics that we were just starting to promote over the past year. And then number two was, a lot of these bleeds may occur, not just during the procedure, but a lot of them are after the procedure, after they've maintained hemostasis. And the biggest thing that we were able to get out of this is by switching the use case of not being an arterial sheath, which you'd eventually pull after the procedure and not be able to monitor. I think the idea that this was actually something that could be used in the femoral vein has now allowed the strategics, which they probably listen to us, but have increased the appreciation of what the utility of the device could be, especially if it could be scaled out beyond just a sheath to monitor these types of bleeds post-procedurally. So yes, there's been, I guess, an increased interest in us just because we've purposefully have promoted, the use of this device in the femoral vein by participating in some of these conferences that I mentioned, specifically at TCT Shark Tank. And since then, we've had multiple meetings face-to-face with the strategics, and what they were asking us is wanting to see the animal study data, which we just completed, a little bit over a month ago. And I think that's, to gain traction. But also, I think they're waiting to see, and this is what's prompting this 2.8 million raise, is how are doctors going to incorporate this kind of monitor, which is, quite new right now, into clinical practice. I mean, obviously they want to see if it behaves the same way as the animal study, but I think they also want to see what the reaction is going to be with those clinicians that are using this in these particular procedures where, there is a perceived risk of bleeding, right? So we're really going to focus on the high-risk TAVR procedures, and we're going to also and the chip cases, so the complex PCIs that have a rate of bleeding, CTOs, and we're going to focus on impellas. The impellas are right now internally, I think they've been talking about rates of bleeds about 25 to 30%. So if we target and show that these devices can actually be able to be integrated into those and they have some value of triggering a detection, we're going to learn from them, from the doctors on how they're going to react to that. I think that's kind of what from my understanding with the strategics, that's what they want to see before they make the move. And that's what
essentially, so as a physician, if they're doing the procedure like an Impella and they detect a bleed or like level 2 or level 3 in which they need to take sort of action for, then I guess the choices are to abandon the procedure or not give anticoagulant, or, I mean, what's the alternative for them?
Yeah, I think there's a number of things that we're going to probably have to learn on how they're going to react to it. I think there could be a lot of, there could be additional diagnostic intervention that they could do to do it early enough to see if they can locate or see where the bleed may be that they wouldn't be able to have done earlier, because not until the patient would become symptomatic would they be able to see or recognize a potential bleed. So it could potentially optimize the actual procedure during the Impella, so if they have to speed it up, change anticoagulants. If they have to reconstitute the vessel while they're in, and I know I've spoken to some interventional cardiologists, they could do a contralateral access and try to reconstitute the vessels during the procedure so as to prevent any potential issue that might be happening. These are usually within the TAVR. With the Impella, I'm not exactly sure how they would do that, with the sheath in there, but there may be an opportunity to do that. But so there's a number of things that we're going to try to capture. But the biggest thing is our original thesis for this particular device is before there is symptoms where you actually even, provide like a unit of a blood for transfusion, you can significantly impact the outcome and then the subsequent costs of the procedure. So whatever they're going to do to be able to kind of, delay, successive blood loss to prevent some of those, transfusions or obviously, other kind of sequelae that might develop from the bleed. That's what we're trying to address. That's how we're going to try to address it with the early monitor.
Okay. All right.
Zafra, is Fred Duffy in Dallas. I can jump in to answer that partially, even though I'm just a plastic surgeon. So sometimes what this will lead to is the need to intervene and get a vascular surgeon involved if these guys can't handle it. But that still shows the value of your device, because the complications of having a big retroperitoneal hematoma, et cetera, infection, skin necrosis, it's significant. So there's real value in that.
Yeah, that's right. I mean, I think there's been conversations I've had where, okay, well, is this going to increase additional costs associated with even like maybe doing a post-procedural CT that might not have happened? Yes, but I think if you kind of outweigh what the potential impact could have been if there indeed was real bleed progression that was occurring that they would not have caught that early, then yes, our budget impact model would state that you're actually probably coming out ahead, even if you have to do an additional diagnostic, or even if it's a consult, like you said, with a surgeon to address an issue.
Correct, and there's not just morbidity or complications and costs. There's deaths with those big bleeds. So.
Okay. And because you're going to be detecting earlier, so you could just, like, for example, if it's that significant bleed, then you would just not give the anticoagulants, which will cause it to bleed even more, or you can just cancel a procedure. Is that correct?
I think those are potential options. I think you could probably, reduce the anticoagulation, see what you can monitor monitor the patient. I mean, I think it depends on how far along you are within the procedure as this comes along. The way the device is going to trigger, you have the first light that would probably trigger at at least in our animal study, about 25 to 30 cc's. But if you see and that might not be that much, right? So the doctor might not even do anything at that point. But until you see that kind of, progressive bleeding, which is indicated by the second and third light triggering on, that's when they would have to make that potential decision, depending on how far they are. Again, I'm not a clinician, but that's kind of what I've been told is they would either try to expedite the procedure if possible, or to your point, do other things, either canceling it or not. But I think the idea of trying to cancel a procedure at this stage with where the device is would be pretty aggressive unless they actually had experience with the device reacting that way. And that's what we're going to try to understand, hopefully, in the first 100 patients.
Okay. And just out of curiosity, but obviously you don't have to comment on this if you can't. But have there been any strategics who have who had said that they were extremely interested in some sort of commitment if the animal studies, if the human studies do show some sort of validation?
They haven't been that explicit, I'll be frank, but they've been highly complimentary of how far and how the device has, number one, on how far we progressed, but also on its utility. I think now that they've been able to better appreciate the use of it, especially as a bleed monitor in its current form. And I think a part of it also has been because we've been able, and what's been nice is, and I don't want to our own horn too much, but we've been able to engage clinicians that have connections to some of these strategics, specifically those that may have bleed issues to begin with. And I think that has kind of, positioned us well, especially when we've had our scientific advisory board affiliated with some of those strategics and have actually been working some of those kind of back channel conversations on, with the utility of the devices. But I'll tell you, as any BD person, and I've been on the other side of things when I was, with a large medtech, we're not going to show our hand yet. They didn't show their hand at this point, but I think what's been tracking is the recognition that we've been getting with the clinicians, and also the progressive level of activity that we've had conversations with the strategics. And we have been invited to one strategic to actually present in person at their headquarters at the end of this month, and then we've had multiple strategics wanting to know, where we were with the animal study data, which we're starting to share now. So I think a lot of it's been a lot more two-way conversations than maybe what we've had in the past. But
okay.
No, there's no term sheet or anything like that on the table yet.
Oh yeah, obviously not. I wouldn't expect there would be. Well, that's good to know.
Zafar, on the subject of exit, and I know Larry is on the phone as well. Talk about just your best estimate on the timing of an exit. How does this play out? And I know the expectations have risen in terms of the potential exit valuation. Give us some sense of what is driving that and some color on it.
Okay. So I think there are some key inflection points that we have coming up that could trigger possible deal making or exits. So I will tell you immediately that, there is something that's coming with, the next few weeks on the FDA submission. That's not necessarily going to trigger anything at this point, but that will set the clock for when an FDA clearance might come up, which we're expecting in the third quarter. So that is, a milestone that if we achieve and table that, and couple that with clinical cases, I think could be an appropriate time for an exit, and even prior to generate any revenue. So that's what we're going to be running towards very quickly, and that's why we're trying to get this clinical pilot up and started. But there could be other exit inflections that could come earlier than that, and that is just getting the first clinical cases in the next quarter. So, we're not going to be shy about sharing our clinical cases with those strategics that we've already signed NDAs with. And as they come along, if there's, cases that help highlight the case, the story that we're trying to demonstrate and see how doctors are using it, we're going to start sharing those as soon as we can once we get the cases. However, we are going to have a large audience in the end of June where we're sponsoring a symposium where we're going to present our initial data, both on our animal study results as well as our hopefully our first clinical pilot cases as well. And I think that might serve really well to kind of tee up, more aggressive conversations with our strategics, and we're going to drive that on our end as well, prior to even getting FDA clearance. So, and then we'll anticipate the pilot to be completed really close around the same time as our FDA clearance coming in. So that's kind of where I see a lot of activity that we're really going to push towards is trying to generate conversation. One of the things that has been happening as of late is there's been multiple interest from strategics and I'll be honest with you, these are device manufacturers. So if we can get interest from multiple strategics, I think the idea of accelerating exit would be helpful, because if we can obviously have a bidding opportunity or showing that our device could complement them in driving share, especially in areas where they may be competing with the strategics that we're speaking with, I think is what we're going to try to leverage strategically. So that's what's driving the timing, and that also is kind of where we feel the valuation actually has increased as well, because when we talk about the device not only supporting one particular therapeutic area, but a number of therapeutic areas, especially with the larger med tech strategics, we feel that the valuation can look somewhere around 150 to 200 million. And we have some samples in that deck that I forwarded on recent exits, and the median exit value has been about 200 million. So it's not unfair to say that we couldn't reach that if we can leverage the data appropriately with what we're seeing, and then leverage the opportunity within the strategics. And like I said, there's large med tech that have multiple therapeutic plays where this could easily fit in and even can scale out with any kind of modifications. The IP, we feel, is very strong in order to allow for that kind of proprietary kind of scaling out in their portfolio. Whether it's, right now it's access site bleeding, but who's to say that it can't be used for bleeding detection in other areas, whether it's higher up in the aorta or coronaries as well. So there's some cool interest, I think, from the conversations we've had with strategics on how this could potentially play out. But those are what the drivers are: the clinical data, and then and the multiple interests that are driving the value. Does that kind of help color the situation a little bit with where our expectations are?
Yeah, that's excellent.
Yeah, quick question: what's the actual cost of the device per kit if you were to include it in a kit? The actual manufacturing cost.
Yeah, that's a good question. It's not a cheap device, right? So I think, and we believe that we can scale, bring down the cost of manufacturing considerably once we get volume. But right now, where we are with what we're having to deal with at this particular stage, is about $200 to build this device.
What? Even at these low volumes, that's not a bad. I guess that's not too expensive compared, when you consider the cost of those kits to begin with.
Yeah, and I think the bottom line is making a case on what our pricing strategy could be, because it's kind of interesting. Love to have a conversation with you guys that maybe associated with some of the purchasing communities at the health systems. We're thinking that we would like to try to price this device under $1,000, and I think we have a good argument to obviously stay wide in terms of what the cost and complications are for the bleed, but also there's another periprocedural safety device out there that priced quite a bit higher for complications that occur less frequently and honestly cost in total less than what the bleeds are costing for some of these interventions that we're targeting. So I think the margins can still be very good. We can probably improve upon that with scale at 200, but we're trying to. I think the biggest thing is to ensure that we can try to get appropriate pricing, maybe around the $800 to $1,000 levels. I don't know what you all think about that, as being clinicians in your health system for a device like this. This is not a sheath. I think that's one of the things that we've learned over the past 5 months. It's a bleed monitoring system that uses a sheath as kind of the conduit, and we want to ensure that people understand that this is, it's not a commoditized sheath just with a few electrodes. It's a comprehensive monitoring system that can monitor the patient beyond just the 1 to 2 hours of the procedure. So.
Well, you're always best, in my experience, to go in as a premium product with premium pricing, at least as the sales pitch to a strategic.
Yep.
What they choose to do after the acquisition is a business decision they have to make. But certainly the ones I've worked with, they consider themselves a premium product provider. And those price points you're talking about would not be exceptional for the kind of thing you're doing. So I think you're on target there.
Thanks. Yeah, as a standalone, I think that's what we would push if we had to sell this. Even, if we have to do a limited launch, that's kind of what we're going to try to position the product as. But to your point, if a strategic incorporated this technology and wanted to bundle this into their, delivery systems of their devices, whether it's a valve or, like a pump, it could be easily absorbed and just be supported as a, an enhanced feature, right? So.
Absolutely.
Great. Any other questions for Zafar? Okay.
I can also follow up on, I don't know if you want me to, I think you want me to mention, Dan, I think the pro rata for the existing syndicate was about a little bit over $100,000 to maintain, the pro rata share as we go into this raise. And I think we've allocated, is that right, $300,000 for angelMD this round?
Is that a question for me?
Yeah, just wanted to verify. So.
Yeah, the number you gave me specifically was $101,366 to maintain our ownership in light of the new raise and the dilution impact. The last number you gave me on allocation for this was $300,000. Of course, we would probably be interested in more if you can allocate more, because I think this would be there would be interest of our Catalyst Fund in looking at this investment as well. So if there's room above that, would like to know ASAP. And so for the current investors in Saranas, I have developed an analysis. There are I believe there are 19 investors, individual investors. I will send each individual investor a note to let you know what the kind of the minimum investment would be to maintain your current ownership in Saranas. But if you would like to invest above that, you are free to do so. And I would ask if y'all could declare what your interest is in investing and how much by the end of the week. That way we can determine of the allocation currently at 300, but hopefully higher, where we stand with our current investors and what excess we may have that we could offer next to our Catalyst Fund, and then after to some outside investors. So, any other questions regarding deal terms, timing, and questions for Zafar? Great. Well, thank you for your time this evening. Zafar, thank you for the very clear explanation. Congratulations on the progress to date. And I will get the communication out to our investors here within the next 24 hours so that they can get back to us.
Great. Thank you.
Right. Thank you.
Good with all. Appreciate it.